Good morning, student. In this video we are going to discuss about the resting membrane potential. At the end of this session you will be able to define the resting membrane potential. You will be able to understand the concept of resting membrane potential, explain the mechanism of genesis and maintenance of resting membrane potential, and you will be able to give the normal values of RMP of different excitable tissues and equilibrium. Now come to the definition of resting membrane potential. So resting membrane potential is nothing but the electrical potential difference across the cell membrane under resting conditions. Okay, so we can, in the other way, we can also say that if we consider this cell membrane, okay, so this is the inside of the cell and another is the outside of the cell. So there is a and one galvanometer is connected over here. You can see the galvanometer is connected over here. So the one electrode of the galvanometer is present inside. It is facing inside. Another electrode, it is on the outside. Okay. So what is happening when we are connecting these two ends with the galvanometer? We can see that there is a deflection of the in the galvanometer. How much is the deflection? It is minus 70 millivolts. So there is a potential difference between the inside and the outside the cell at the resting state when there is no excitation, no stimulation. So this potential difference between intracellular space and the extracellular space at the resting state is known as the resting membrane potential. So for definition you have to remember three things. It is a potential difference between what? Between intracellular and extracellular space or across the cell membrane you can say. And when the difference is there, it is at the resting state. So this is called the resting membrane potential. The normal resting membrane potential values of the excitable tissues, that is the tissues that can after stimulation we can change the polarity. For example, the neurons, in case of neurons it is minus 70 millivolts. In case of skeletal muscle it is minus 90 millivolts. In case of cardiac muscle it is minus 90 volt. Cardiac muscle can be again subdivided into working myocardial cell and the pacemaker cell. In case of working myocardial cell it is minus 90 millivolts. Now one important concept I want to highlight over here, that is all the cells in our body, they are polarized. Polarized means they have a intracellular negativity. You can say that it is intracellularly it is negative and extracellularly it is positive. Okay, and the deflection and the difference is, it is say minus 70 or minus 80 millivolts. This is at the resting state. Okay, if the tissue is excitable, then if we give a stimulus to this tissue or cell, they can change their polarity. How can they change? If the intracellular negativity is changed to intracellular positivity, you can see that if the intracellular part it is becoming positive, so the deflection it is now plus 40 millivolts. So when intracellular negativity is changed to intracellular positivity, that term is known as the depolarization. Whereas if the intracellular negativity is further increased, okay, so you can see here the deflection, initially it was say minus 80, so it is say minus 100, minus 90, it is becoming more negative inside. That means that will be known as the hyperpolarization. Okay, so normally all cells are intracellularly negative and extracellularly positive. In excitable tissue, if we give stimulation to them, they can change this polarity. Either they can be depolarized, that is intracellularly they will become positive. So depolarized state is the active state. Depolarized state is the active state. Whereas normally what we see, that when the cell intracellular negativity is more, that is the cell is hyperpolarized, it will become inactive. So this is about the polarity at the resting state, depolarization and hyperpolarization. Now what are the causes of intracellular negativity? Intracellular negativity, we can discuss it among the by these 5 points. Why there is more intracellular negative charges? We can explain it by 5 explanations, by 5 points. Number 1 is the selective permeability of the cell membrane. Second point is the His-Donan membrane equilibrium. Third is the Nernst equation. Fourth is the constant field Goldman equation. And fifth one is the sodium-potassium ATPase pump. By these 5 points, we can explain why cell interior is negative and cell exterior is positive, and it is maintained in the resting state. So come to the first point, that is the selective permeability of the cell membrane. What it is? Selective permeability means that in the membrane means that the cell membrane has some control over what can cross it. Okay, so that only certain molecules either enter or leave the cell. So cell membrane is partial, it has got some partiality. It will allow some particles to move in, whereas it will prevent the movement of other particles. So that is known as selective permeability. For example, if we look into this picture on the right hand side, you can see the small non-polar molecules like oxygen, carbon dioxide, nitrogen, steroid hormones, they can pass very easily. All of them, they will face no hurdle or barrier. They will pass very easily. Second thing is the small uncharged polar molecules like water, ethanol, glycerol. Some of them it will go, but most of them will be reflected back. Whereas larger uncharged polar molecules like amino acids, glucose and nucleosides, the very small amount of it will enter. Most of it will be reflected back. Especially in case of ions, if you look into the ions, sodium ion, potassium ion, chloride ion, magnesium, bicarbonate, you will see that they will not be able to penetrate the cell membrane. These polar things, all of them will be reflected back. All of them will be reflected back. So this is the selective permeability of the cell membrane. Now how it is causing the intracellular negativity, we will see later. So what is Donan effect? Donan effect means when an ion on one side of a membrane cannot diffuse to the membrane, the distribution of the other ions to which the membrane is permeable is affected in a predictable way. What does it mean? Because suppose there is one ion, suppose this is the ion, it is not diffusible. So this part of the segment also has got some positively charged ion, some negatively charged ion. So this ion cannot diffuse. Okay. Now it will affect the movement of these other particles, other diffusible ions across the cell membrane. Okay. How? Suppose this ion is a negatively charged ion or anion. So obviously it will prevent the movement of the diffusible cations or positively charged ion, because it will attract it, whereas it will facilitate the movement of this negatively charged ion to opposite side, because it will repel it. Okay, so some ion particles their movement will be favored, some particles their movement will be hindered. So these non-diffusible ions, it will affect the movement of the other diffusible ions in a predictable way. So this effect is known as the Donan effect. Now consider this situation. At the central portion you can see here. So there is a membrane N, and both the side, on the other side, one side of the membrane we have the X compartment, on the other side of the membrane we have the Y compartment. Okay. Now this membrane, it is impermeable to charged protein. That is, the protein molecule, it is impermeable to the membrane, whereas the membrane is freely permeable to potassium and chloride. So what will happen initially? Initially the chloride will move along its concentration gradient to the compartment X. Okay. So what will happen, that the negatively charged particle will be more on this X side. So it will draw or attract the positively charged potassium on the other side. Okay, so initially the potassium concentration, say it was equal on both the sides, but as the chloride is moving along its concentration gradient, so it will draw the potassium. So when the potassium will draw it, so more osmotically active particle will be here. So we will see that more potassium is present on the side A, and at the end we will see that more osmotically active particles are on compartment X, that is the X will contain more potassium, chloride and protein, whereas Y will contain only potassium and chloride. So more osmotically active particle on the X compartment. Okay, so the Donan and Gibbs showed that in presence of a non-diffusible ion, that is in this case it is the protein, the diffusible ions distribute themselves so that at the equilibrium their concentration ratios are equal. Ratios of positively charged permeable ions, it will be equal to the ratio of the negatively charged permeable ions. So positively charged potassium ions on the side X and Y, their ratio will be equal to negatively charged chloride ion on the X and Y, it will be equal. And if we cross multiply, we will see that it is potassium in the X compartment and chloride multiplied by chloride in the X compartment will be equal to potassium in the Y compartment multiplied by the chloride into the Y compartment. This is known as the Gibbs-Donan equation. It holds for any pair of cations and anions of same valence. Here we can take a simpler scenario. So before the equilibrium, you can see that at one side of the compartment we have one equivalent of concentration of potassium, on the other side one equivalent concentration of potassium. So the potassium concentration was equal. So but the chloride concentration, it was more on the other side, and the protein was also there, and this protein is impermeable to the cell membrane. Okay, so this protein is impermeable. So what will happen first? First the chloride, it will move along its concentration gradient, because it is more concentrated on the one side, so it will move in this direction. So when more negatively charged ion is present at this compartment, so obviously it will drag. Okay, so the inside of the cell will become more negative. So it will drag the potassium, and potassium will move down its electrical gradient. Okay, and as osmotically active particle will be more, so it will draw the water, and there will be more water over here. So at the end of this, when an equilibrium is there, you can see that the potassium concentration, it is more than one equivalent in the one side, that is inside, and it is less than 1. Initially it was 1 at the outside of the cell. So potassium concentration, it is increased. Okay, and the chloride concentration is also increased. So osmotically active particle, it is more in the inside. So there is unequal distribution of the ion. So when what we have said before, that what are the factors that responsible for this intracellular negativity? One was the selective permeability that we have seen here. This protein is not permeable. Second is the Gibbs-Donan effect. So the ion which is not diffusible, it will affect the movement of the other diffusible ion, and that will cause the unequal distribution of the ions. Okay, so this is one point. Here also one example, here a value is also given. So initially the potassium and chloride concentration, chloride was more on one side. So initially the chloride will move first, then it will drag the potassium, then it will drag the water. So what will happen at the equilibrium? Potassium ion, which was initially 120, it will become 160. Chloride was totally absent, its concentration will be increased, whereas on the other side both of them will be decreased. So we can say that at one side osmotically active particle will be more, but Gibbs and Donan showed that the ratios of these diffusible ions, it will be same, and if we cross multiply, the potassium of inside and chloride of inside will be equal to potassium of outside and chloride of outside. So in this way, so we have this is called the Donan effect, which is responsible for the unequal distribution of ions. So the effect of this Dunan effect on the distribution of ions has got three major effect on the movement. Okay. First is because of charged protein in cells, there are more osmotically active particles in cells than in the interstitial fluid, and because animal cells have flexible walls, osmosis would make them swell. We know that when the chloride will come inside, it will drag the potassium, then more osmotically active particles, so water will come along with it. So intracellularly there will be more water, so the cell will be swelled up and eventually it will rupture if it were not for sodium potassium ATPase pumping ions back out of the cells. Okay, thus normal cell volume and pressure depends on sodium potassium ATPase. We will explain this first point, the effect of this Gibbs-Dunan equilibrium. Three effects. The first effect is this, we will discuss it later. Come to the second point. Because at the equilibrium, this is very important, the distribution of permeant ions, the diffusible ions across the membrane is asymmetric and electrical difference exists across the membrane. So we told you that Dunan effect will cause what? The unequal distribution. And due to this asymmetric distribution, there will be an electrical difference will be created across the membrane, and the magnitude of this electrical difference can be determined. The individual ions magnitude of the electrical difference of these individual ions at the equilibrium, it can be calculated by the Nernst equation. Now we are going to explain it and before that, just come to the third point. Third, because there are more proteins in the plasma than in interstitial fluid, there is a Dunan effect on ion movement across the capillary wall. So these are the three effects. First one, it is telling about the function of sodium potassium ATPase pump. Third one, Dunan effect of ion movement across the capillary wall, it is talking about. Second point is very important. This unequal distribution is creating an electrical difference, potential difference, and at the equilibrium, this potential difference of individual ions can be calculated by the Nernst equation. Now we want to know what is Nernst equation. So what is Nernst equation? So this is the Nernst equation where we see that dk is equal to minus RT by ZF log base E ion concentration inside ion concentration outside. If we take it the ion as a potassium ion, so potassium concentration inside, potassium concentration outside. Okay, so Vk is the equilibrium electrical potential difference. So this is the equilibrium potential that we are determining by the Nernst equation. Equilibrium potential of the diffusible ion, that is what are those ions which are diffusible? Sodium, potassium and chloride. So Nernst equation will tell us about the equilibrium potential of potassium, equilibrium potential of sodium and equilibrium potential of chloride. Minus, this is RT, R is the gas constant 8.31 joule per kilomole, and T is the temperature in Kelvin, divided by ZF. Z is the valency of the ion. In case of potassium it is +1, and F is the Faraday's constant 96,500 coulombs per mole, whereas it is log base E and potassium concentration inside and potassium concentration outside. If we reverse it, potassium outside by potassium inside, so it will be negative log, so the negative sign will be positive. So this is the Nernst equation. Now we want to know what exactly it is telling and how we can derive it. Now think about a situation. So here we have one membrane. This is a semi-permeable membrane. On each side we have two compartments, compartment A and compartment B. Okay, and we in the compartment A we have X positively charged ion X, its concentration is 1 mole, and in the compartment B we also have the positively charged particle X, whose concentration is 0.1 mole. So from here we can see that the X particle is more on side A. Okay, so what is the normal tendency? Along the concentration gradient, obviously the positively charged particle, it will move from compartment A to compartment B. So this is a normal tendency. Now if we are now adding some negatively charged ions, okay, if there is no potential, electrical potential difference, so between side A and side B, so from side A the X will move to side B. Okay, now we are making that side A more electrically negative. Okay, so if more electrically negative A, compartment A, so do you think it will allow the positively charged potash X plus ion to move to the other side? No, it will try to attract it. It will try to hinder the movement of X plus. So as long as there is no electrical gradient, it is okay. Along the concentration gradient, smoothly the X from higher concentration to lower concentration, it is moving. But if we make the compartment A electrically negative than compartment B, so what will happen? So the direction of net X plus movement, it will depend whether which one is dominant, concentration gradient or the electrical gradient. The one who is dominant will actually control the movement of X plus. If the concentration gradient wins the war, then obviously X plus will move from A to B. If the electrical gradient wins the war, so it will not allow the positively charged X plus ion to move from A to B. Okay, so the direction of net X plus movement, now it will depend upon these two things, both the concentration difference as well as the electrical potential difference. Now by comparing these two tendencies, concentration and electrical, we can now predict the direction of net X plus movement. Okay, so we can say that the quantity here, we can say the quantity that allows this comparison. How we are going to compare that who will win? Okay, the quantity that allows us to compare the relative contributions of the ionic concentration and the electrical potential to the movement of an ion is called the electrochemical potential. So this one is your electrochemical potential, this one. Okay. Electrochemical potential means what? Is difference in energy that exists on a mole of X. On this mole of X, the difference of energy that is existing on it and the side A, and the mole of X plus ion on side B from both the concentration and electrical potential difference. So in the side A, on the X plus we have the effect of concentration and electrical gradient. On the side B, on X plus we have both the effect of concentration and electrical potential difference. The difference between these two will actually define this electrochemical potential, which is equal to this we can say the mu A X plus, that is the concentration gradient, energy due to the concentration gradient, plus the mu B X plus, that is energy due to the electrical potential difference. Now this energy due to concentration gradient or tendency to move from A to B, okay, because of the concentration gradient. So concentration gradient, how can we calculate it? We calculate it is the RT concentration gradient means the concentration difference, that is RT log n concentration of X in the A divided by concentration of X in the B. So this is the concentration gradient. And energy due to the electrical gradient or tendency of ions to move A to B because of the electrical potential difference, you can see here, so that is equal to ZF into electrical difference between the potential difference between the compartment A and compartment B. So we can say the value of mu A is this RT ln and this, and value of this electrical difference is ZF minus. So this is understandable. This is known as the electrochemical potential. Now after this hassle and pull and push and everything, who will win, who will not win. After certain time there will be an equilibrium. Okay, so at the equilibrium, after all war, so there will be distribution, the ions will come to a peaceful state. Okay, now what will happen at this equilibrium state? This concentration gradient, energy due to concentration gradient plus energy due to electrical gradient will be equal to 0. Okay, and from that we can derive the Nernst equation that we will see in the next slide. So this is the another mathematical form of the previous discussion. Electrochemical potential energy is equal to the chemical potential energy difference plus the electrical potential energy difference. Okay, so it is actually RT into the concentration of X in the compartment A minus concentration of X in the compartment Y, inside or outside. So we took the logarithm of it. That's why it is Xi divided by Xo. As the concentration is very less, so we took the logarithm of it. Okay, so it is the RT into concentration difference that we took the logarithm plus ZF into the electrical potential difference, and it is equal to the electrochemical potential energy difference. Now at the equilibrium we know that this RT log n Xi by Xo plus ZF into the A minus EB or phi minus phi O is equal to 0. Okay, so if it is 0, so that means we can say that A minus EB, if we derive this equation, we can find out in this way that initially it was ZF into A minus EB, it was ZF into A minus EB plus this RT by ZF log XA by XY, it is equal to 0. So we took the RT by ZF on the other side, so that's why it has become this minus RT by ZF log this. Then EA minus EB will be RT and ZF will come down. So here it is the membrane potential. We can say that this potential difference is actually the potential difference between inside the outside the cell at the resting state. So it is the equilibrium potential, membrane potential for a specific ion, and R is the gas constant, T is the absolute temperature, Z is the valency of that ion, S is the Faraday constant, log base E in an intracellular concentration of that ion divided by extracellular concentration of that ion. Okay, so in this way, this is the equation we have derived the this is the Nernst equation we have derived. How we have derived? So first we considered the concentration gradient and electrical gradient on the ion. That sum of them will be 0 at the equilibrium. So from that we can derive the equilibrium potential E. Now we want to know the equilibrium potential for all these diffusible potential for all these diffusible ions, that is sodium, potassium and the chloride. Okay, so what is that? So before that we will convert this log base E into log 10. If we convert it, then we have to multiply it by 2.303, that we know. Okay, so we will EA minus EB, so we will multiply this minus 2.303 RT divided by ZF or NF, and log 10 log base 10 concentration of X inside divided by concentration of X outside. Then we know that this RT and the S, it is also constant. If we for all practical purposes, R is 8.313 joules, F is 96,500 coulomb. Therefore at 298 Kelvin, so we can say the RT by S, 2.303 RT by S will be equal to 0.592 or around 0.61 or 0.6. This value will come. So we can say the potential equilibrium potential will be minus 0.61 divided by the valency, that is N or F, we can say is into log 10 concentration of an ion inside or outside. Now, so if we know the valency of this ion, suppose in case of potassium, we know the valency is +1. if we know the concentration of potassium inside the cell, if we know the concentration of potassium outside the cell, so we can easily derive the equilibrium potential of potassium. That is, when the membrane is only permeable to potassium, how the potassium will move, how it will drive the resting membrane potential, that we will get understand. So if we look into here, so here one example is given that potassium, it is inside, it is 10 millimeter of mercury, and it is outside 1 millimeter of mercury. We know that intracellularly potassium is more, so outside it is 1 and inside it is 10. So 1 by 10, that is minus value, so minus into minus, it will become positive. So it will okay, so here they have already altered, that is initially minus RT by ZF log concentration of potassium inside by outside, so they have reversed it, so the negative charge they have taken out, so it was outside by inside, so outside by inside, now it has become 1 by 10, so that's why again it is minus log 1, so the equilibrium potential, it has become minus 58 millivolts. So similarly we will see here, so we can say that if we make in these compartments, okay, so we know that potassium is very high at compartment A, so along the concentration gradient it will try to move on the potassium compartment B. Now if we increase the negativity, how much? 58 times, or it will be 58 millivolt more negative relative to the side of side B, 10 times more. So it is, as the concentration is it is 10 times more, so if 10 times more negativity should be there, or it has to become minus 58 millivolts on the compartment A to prevent any movement of potassium from A to B, and the equilibrium will set in. Okay, so concentration gradients, normally it will move. Now if we want to prevent this movement, we have to make this compartment A electrically negative. How much? Minus 80 millivolts should be the value. So we can quantify how, by the Nernst equation, what should be the electrical value by which we can reach the equilibrium. So in this way, if we know the concentration of different ions, the concentration of potassium and chloride inside and outside, and we know these values, and we know the valency, we can calculate their equilibrium potential. Okay. So what is the equilibrium? We know the values, intracellular and extracellular concentration of sodium and potassium and chloride. So if we put their value, we will find that the equilibrium potential of potassium, it comes around minus 90 or 96. Equilibrium potential of sodium, it becomes plus 52, or in this case it is plus 52, but ideally it is plus 65 millivolts. Okay, and in case of chloride it will minus 80 millivolts. Okay, so this is the equilibrium potential. What does it signify? Now suppose this membrane is only permeable to potassium. Okay, so potassium will try to drag what is the normal resting membrane potential? It was minus 70, I think. You remember, it is minus 70 millivolts. So if the potassium is allowed to move, okay, so what it will try to do? It will try to drag this resting membrane potential to its equilibrium potential. How much it is? Minus 96. How can we make it minus 96? By making more intracellular compartment more negative, that is hyperpolarizing it. So the tendency of potassium is to move outside. So when the potassium, positively charged potassium, is moving outside and outside and outside, how long it will move? Until and unless the resting membrane potential touches the value of its equilibrium potential, that is say minus 90 millivolts. Same case in case of sodium. What is the value of it? It is say plus 65. So if only sodium is allowed to move, what will be the effect movement of sodium? Sodium will try to drag this resting membrane potential minus 70 to plus 65. That means intracellularly it will try to make more positive. So what will be the movement? In which direction it will move? The positively charged sodium will try to move inside. It will try to depolarize. So it will move inside on and on and on. How long? that when this resting membrane potential will reach this value of plus 65 millivolts, it will continue to move. Okay, so all these equilibrium potential is actually giving an idea if these ions are given free hand in which direction they are trying to drag the resting membrane potential. In case of potassium, it will cause more hyperpolarization. In case of sodium, it will cause depolarization. So in this picture, we can clearly see that this potassium ion, their tendency is always try to potassium ions through the potassium leak channels, they are trying to go outside and trying to drag the resting membrane potential to minus 90 millivolts, whereas sodium ions through their leak channels, they are trying to move inside until and unless the intracellular positivity touches equilibrium potential of plus 60 millivolts. Okay, so but it is an ideal condition when we are allowing only potassium, allowing only sodium. But normally what happens, all these sodium and potassium, they are moving simultaneously and they are pushing and pulling each other, and after certain time, when there is an equilibrium is reached, okay, so we will get a resting value of potential difference between inside and outside, where the inside will be negative and outside will be positive. So that due to the Donan effect, unequal distribution, and this unequal distribution will generate a potential difference that we quantitatively measure by the equilibrium potential of individual diffusible ions. What are those? Potassium, sodium, and the chloride. So from the Nernst equation, we came to know about the equilibrium potential of potassium, equilibrium potential of sodium, and equilibrium potential of chloride. And from it, we will try to derive how can we reach the value of resting membrane potential by these individual equilibrium potential. Now, we know that 1 ampere current means 1 coulomb of charge flow per second. Okay. Now, each monovalent ion, any monovalent ion, say potassium, one potassium positively charged ion, it will carry 1.6 into 10 to the power minus 19 coulombs of electrical charge. So we can say in the other word, 1.6 into 10 to the power minus 19 coulombs it is equal to 1 potassium ion. So 1 coulomb current will be equal to, we can derive it that 1 by 1.6 into 10 to the power minus 19. If we derive it, we will get the 6.25 into 10 to the power 18 potassium ion. Okay, so if 6.25 into 10 to the power 18 potassium ion, they flow per second, okay, across the cell membrane, it will create 1 ampere of potassium current. This is clear. Now we want to know about the current density. Okay, actually this current is moving across a membrane. So membrane has a surface area. Okay, so the total what is current density? The total flow of charge per time, that is the flow of charge, that is current per time, that we know over a cross sectional area of cell m square. So that is the that is known as the current density. And if we consider the current density, this current density of the membrane can be calculated by Ohm's law. We know that Ohm's law is equal to V is equal to IR, where I is the current and R is the resistance. Okay, V is equal to IR. So current, we can say I is equal to then V by R. So V into 1 by R we can say. Now 1 by R is nothing but the conductance or G. So G is equal to 1 by R. So we can say that I or current is equal to the the voltage into voltage difference into the conductance of that ion. So from this we can derive the outer current per unit area, that is microampere per centimeter square. So how can we calculate the current? This potassium flowing outside, potassium is moving outside, this charged particle is moving outside across a cell membrane which has got a surface area. So how can we calculate this current across this membrane? we can calculate it by the membrane potential minus the equilibrium potential of that ion. For example, the membrane potential of say we know in case of cell it is minus 70. In case of potassium, the equilibrium potential is minus 90 into the conductance of the potassium. So by this we can calculate the current created by the potassium ion. Similarly, we can calculate the current of sodium. So potassium movement is outward, so we can calculate the outward potassium current. Similarly, the inward sodium current and the chloride current can also be calculated. So in this way, we can say that the sodium current we can derive the membrane potential minus the equilibrium potential of sodium, that we know plus 65, into the conductance of sodium. Okay. Now similarly, in case of potassium, the membrane potential, resting membrane potential minus the equilibrium potential of potassium. From Nernst equation we got it minus 90 into the conductance of potassium, and the chloride current will be equal to the resting membrane potential minus the equilibrium potential of chloride into the conductance of chloride. So the potential at which the equilibrium is established between the depolarizing and hyperpolarizing current, it will denote the resting membrane potential. So for this, you just remember that at the equilibrium, this I current, the current for potassium, sodium and chloride will be equal to 0. And we know how can we derive individual currents, sodium current and potassium current. We will put their value. So this is the value of potassium current. Okay. So this is the value of sodium current. This is the value of chloride current and calcium. So usually we take these three, and if we derive this equation, we will get the EM or value of membrane potential. So from the Nernst equation we have derived the equilibrium potential, and from that we have derived the individual current, and by multiplying the difference with the conductance. And if we plotted this value, so at the equilibrium we know there will be no current, all these currents will be equal to 0, their sum will be equal to 0. So if we put this value, we will give this equation, chord conductance equation, from which we will get the value of membrane potential. That we already know. In the neuron it is minus 70 millivolts, in the skeletal muscle it is minus 90, in case of working myocardial cell it is minus 90 millivolts, that we have derived. So this is the chord conductance. So we know that membrane potential, that I have told in the previous slide already, that membrane potential is equal to the conductance of potassium into the equilibrium potential of potassium plus conductance of sodium and equilibrium potential of sodium, conductance of chloride and equilibrium potential of chloride, calcium of that, divided by the conductance of all these, the total conductance, and we will get our coveted value of resting. By mathematically we have calculated this one. Okay, now we will see. So it will say that it is minus 70 millivolts. Now we will individually plotting it. We will try to see this value, whether actually it is, we have calculated it, whether it is actually it is minus 80 or minus 70 millivolt or not. Suppose take a condition where we say that no, the membrane potential difference, there is no difference, it is equal to 0. It is not minus 80. Okay. So we know the equilibrium potential of potassium, it is minus 90, and we have measured the value of conductance of potassium, it is 0.6. And we know the membrane potential of equilibrium potential of sodium, say it is plus 70, and we know the conductance value of sodium is 0.06. And if we know this chloride, it is minus 80, and the conductance of chloride is 0.3. Okay. So from it, if we plot this value in this formula, so we get the individual current. Okay. So here the current value for potassium, it becomes 54, and we know the direction of current. It is from intracellular, it is the outward current. Okay. So it is plus 54. Okay, we have got this value. It is a hyperpolarizing current. That is intracellular, LD, it will become more negative. Okay. And what about the chloride? Chloride is also, it is a hyperpolarizing current, and the current value is say 24. But only depolarizing current is here, it is sodium, it is 0.8. So two currents, so potassium current and the chloride current, both these current are hyperpolarizing current. And what is their strength? That is 54 plus 24, we can say it is around 78 microampere. So total hyperpolarizing current is it is 78, whereas only near 2.8 microampere current is trying to making the intercellular positivity, that is sodium. So which power is more dominant here? The power that is causing hyperpolarization, that is intercellular negativity. So we can say that it is not the zero. Obviously the intracellular part should be negative. How much negative? When the hyperpolarizing and depolarizing current will balance each other, obviously that should be the point. So sodium current is the only near 2.8 depolarizing bond. The other two demons are the chloride current and the potassium current. They are hyperpolarizing current and trying to make the intracellular part negative. How much negative? When the depolarizing, so it will become negative from 0 to say minus 10, minus 20, minus 30, minus 40, like this it will go on and on. At a point when the depolarizing current will be equal to the hyperpolarizing point. And what is that point? So suppose now take the membrane potential minus 80, and these rest of the values are same. Now calculate the current. The hyperpolarizing potassium current is now plus 6. The depolarizing sodium current is now minus 6, and the chloride it becomes 0. Okay. So with increasing negativity, the depolarizing sodium current increases and hyperpolarizing currents of potassium and chloride decrease. So when membrane potential is minus 80 millivolts, the potassium will carry toward hyperpolarizing current of 6 millivolts. Sodium will carry inward depolarizing current of 6 millivolts. So they will balance each other. So total hyperpolarizing will be equal to the total depolarizing current, and the membrane potential will come to a rest. So that we can also prove by this equation that membrane potential it is resting membrane potential it is minus 80 millivolt, and intracellular part it is negative. Now these current, we know the rate of diffusion of ion is current. These current can be of two types. One is a channel current, that is happening through the leaky channels of potassium, sodium and chloride. Okay. Another current can be pump current, that is done by the sodium potassium ATPase pump. But so I told you when in the first point, what are the points responsible for the intercellular negativity or resting membrane potential? One is the semi-permeability, then the Donan effect, then the Nernst equation, then we have the Viscount's conductance equation. So all we have derived, and last point was the sodium potassium ATPase pump. So sodium potassium ATPase pump actually at the resting state, it works very feebly. So it has very less, non-significant effect on the resting membrane potential, on the creation of resting membrane potential. Then what is the function of it? That already we have to discuss. The function of sodium potassium ATPase pump is to maintain the cell volume. So when there is unequal distribution of ions due to Donan effect, so one side intracellular part of the cell will have more osmotically active particles, more water particles, so cell will swell up, it will try to rupture. So that will be prevented by sodium potassium ATPase pump, but it has got very mere effect on the generation of resting membrane potential. So we know the definition of resting membrane potential. We have selective permeability, how it is affecting, Gibbs Donan's membrane equilibrium and Nernst equation, constant field Goldman equation or conductance equation, and the function of sodium potassium ATPase pump. So thank you. So this is the reference.